All right, let's go ahead and get started. Before we get started the lecture, one quick announcement. We're gonna have our first assignment due next week. This is gonna be the beginning of the semester-long project. The first assignment is gonna be just identifying what the biological problem or disease that you want to focus on. It's gonna be it's gonna be a short paragraph that's identifying what that disease or biological problem is. Um, the information on this semester-long project and the different aspects of it are both in the introductory lecture recording as well as in the syllabus. But the idea is that we're going to be focusing on some disease or biological problem and focusing on it from a cellular or molecular biology. We're going to view it through a cellular molecular biology lens. Um, for a lot of these, you're going to be either trying to focus on identifying potentially a new and better alternative therapeutic approach, or potentially identifying a new potential mechanism if there's not a clear pathogenesis for the disease. Um, I know some of this might seem really daunting, but the idea is uh, to try and give you guys license to come up with some really big ideas and to go into the literature and to try and read some journal articles about this. There are different kinds of articles that you can read when you're jumping into literature. There are either research articles, which are very specific studies that groups have done and reported their findings, or there are review articles. Review articles will give you kind of a landscape of the field over the course of the last maybe 15 or 20 years or so. Um, you want to focus a lot of your reading on research articles. Um, uh, Review papers are going to give you they're going to help give you a sense of what the landscape is, like what's known and what's unknown. But those are not going to be the main things you want to focus on. That'll help, again, it'll help give you bearings, but you don't want to rely all of your reading on review papers. Try and focus on research articles. They're going to give you a sense of what are some potential mechanisms or therapies that do work or don't work. And but for the first assignment, again, you're going to be focusing on just identifying the disease or identifying the biological problem you want to focus on, and you're going to want to articulate what the molecular biology or cell biology lens through which you're going to focus on. I'm going to provide details for the assignment when I post it later tonight. It's going to be due next Tuesday, so you have a week to think about this and spend some time reflecting on it. For some of you, you may already have an idea of something that's really caught your attention that you feel really strongly about, that you might have a personal connection to. For some of you, there might not be a clear thing that comes to mind, and that's okay. Um, if you want to try and look for inspiration on what you could focus on, you can actually look at some of the research that's going on in the department right now. There's a lot of work going on in our department that falls under the category of cellular molecular biology. And so looking at some of the ongoing work in the department can help perhaps spark some ideas or give you some inspiration. Are there any questions about that at all? Okay. And so as we're working on this, there's going to be just multiple small assignments to kind of keep you guys on pace rather than just giving you one big assignment at the end of the semester that not all of you, some of you might be tempted to just push it back and just save it for the very last week of the semester, which can be quite daunting. But we're going to have just like small assignments to help give you feedback and keep you on track. And there's going to be three main deliverables that we're going to focus on for this semester for this project. There's going to be a figure illustration that you create, and I'll give you access to a really helpful piece of software that will that will enable you all to make some really compelling images. But a figure illustration that communicates this idea, we're going to work on creating 3-minute theses, just like a 3-minute video that communicates what the problem is and what your idea is. And then you're going to write a 2-page research statement. It's going to fit the format of the Harrison Undergraduate Research Award, as well as the NSF Graduate Research Fellowship. Um, Particularly for those of you who are third years and fourth years, the NSF Graduate Research Fellowship is typically a really competitive fellowship that if you are awarded it, it can get you a ticket to any PhD program in the country. We've actually had a few students who've gotten that award that were in this class when they received it. I'm not saying it's because of what they did in here, but because they got it, I can say they were in this class when they got it. Um, but again, it honestly is the kind of thing that will enable you to punch your ticket to any graduate program in the country. And so again, it's just 2 pages, and it's actually quite brief. And so the challenge is not trying to fill out 2 pages, but communicate all your ideas within the constraints of 2 pages. And so there's the idea is that you're going to be giving feedback to each other. We're going to have like little study sections of people who are in similar domains of research that you can kind of leverage one another's strengths to give each other feedback and communicate your ideas through different mediums and help you guys develop the skills to communicate your ideas in compelling ways. Um, I could talk about the research project a lot longer, but I just because that's usually all communicated in the introductory lecture, and I only provide a recording of that. I felt compelled to at least give a small summary of it um, during lecture. Are there any questions about that at all? All right. With that out of the way, we'll go ahead and jump into our content for today. So we spent last lecture jumping into the cell membrane and discussing some of the different proteins that are on the cell membrane. And the first lecture was discussing what proteins are and what amino acids are. And so today we're going to talk about a very specific class of proteins called enzymes, and we're going to be talking about them within the context of botulism. So botulism is caused by the bacterium Clostridium botulinum. That is a spore-forming bacterium. You've probably heard about this before, but spores are typically a way in which bacteria can protect themselves. That is a form they can take by which they can protect themselves from external environmental factors, whether it's, for example, temperature. And so this is a pretty common soil bacterium. In addition to that, it's not only found in poorly kept soil, but it's also commonly found in improperly stored food. Uh, and there's one particular food that is at risk of having the spores of this bacterium. Can anyone think of a food product that might be at risk of having particular bacterial spores in them? Yes? Rice, certainly, but there's one in particular I'm thinking of that is problematic for certain populations of people. Yes? Honey, that's exactly right. Particularly locally sourced honey. There's a lot of health benefits of locally sourced honey. For those of you that have allergies, there's been a lot of studies that have shown that, as an adult, if you eat local honey, you'll develop a better tolerance for these local allergens that you're typically exposed to. This is one arm of like ongoing work in allergies and asthma that local honey can have a really great benefit in that way, but particularly for newborns, toddlers, and pregnant women, raw honey can have a fair amount of Clostridium botulinum spores within them, and so that's something to be mindful of. And so, again, it's typically found in poorly stored food, honey. It can be found in poorly cleaned wound beds as well as in dirty soil. The symptoms of botulism poisoning is flaccid paralysis, this idea to not be able to control or contract the muscles. And you see here in this image, there's flaccid paralysis of the face here that leads to facial weakness. There's oftentimes difficulty swallowing or speaking. There's also drooping eyelids. There's also dyspnea, this difficulty breathing, the difficulty of contracting the diaphragm. And in addition to that, there's also nausea, vomiting, and abdominal cramps. If you look at this list of symptoms, you'll notice that there's kind of a common thread through a lot of these. That there's a lot of neurologically associated symptoms, which actually ties to the actual mechanism by which this bacterium and its associated toxin works. Typically, botulism poisoning is pretty rare. There's only hundreds of cases typically within the US on an annual basis. The key way in which the bacterium works is that it secretes a specific toxin, the botulinum toxin. And in case you might be wondering, that's also the same toxin that's used in Botox, but just used at very, very low doses. It does occur, but very, very rarely instances of botulism poisoning after a Botox injection. But what's really important to note is that among the different toxins that we've characterized, it's among the world's most deadly toxins. The way in which we characterize that is by looking at the LD50. Does anyone want to help us define what LD50 is? You want to go for it? That's exactly right. It is the lethal dose, LD, lethal dose, at which 50% of some population of people or animals in a study would die. And so the LD50 for botulinum toxin is about 100 nanograms for an average person or an average adult. For context, I imagine it's hard to appreciate that outside the context of perhaps other toxins, but for context, this is about 4,000 times stronger than cyanide. And so it's really it's because of the LD50 we're able to you can compare the LD50 of other toxins or other reagents and be able to compare their toxicity levels based on that value. And the reason why we're talking about botulism in this enzyme lecture is that this botulinum toxin itself is an enzyme. And so we're going to circle back to botulism poisoning at the end to understand how how this toxin works as an enzyme. Before we jump into our pivot to our enzyme content, are there any questions here about botulism? Yes. Yeah, the way I mean, this is gonna sound pretty grotesque perhaps, but the really because that value is defined by 50% of some population dying, you would likely have to do an animal study where you will use different doses for different populations of subjects and see at this dose how many subjects die, at this dose how many subjects die, and at this dose how many subjects die. And at certain doses, you'll be able to determine at which dose is about 50% of them dying, if that makes sense. Timothy? Yeah, that's a great question. So, right, yeah. And so because of that, we usually generally talk about LD50 as 50% of some population. And so in an animal, as you can imagine, in a mouse that weighs 20 to 30 grams, whereas the average person weighs somewhere between 60 to 80 kilograms, that dose is going to differ dramatically. And so there's going to be a different LD50 for a 25-gram mouse versus a 70-kilogram adult. And so you define that differently based on the dosage. We're not doing randomized clinical trials determining LD50s for toxins. That will typically just be determined, I think, usually based on exposure, if that makes sense. People more observationally rather than as a research question with animal subjects, if that makes sense. All right, with that, oh, yes. Yeah, so it's a great question. The noted, we'll talk about, what's a great way to say this without jumping too far ahead? Uh, Being mindful of the symptoms that we just talked about, these like neurologically associated symptoms. Um, there is that flaccid paralysis helps reduce wrinkles, for lack of a better phrase. And so there's a cosmetic benefit to it as long as it's used below a certain threshold that doesn't jeopardize the safety of that patient. Um, it is, I would say, not giving life advice here, potentially unwise to expose yourself to a toxin like this that is 4,000 times stronger than cyanide, but um, that is each person's prerogative. But yeah, I think it's just the noted effect on local nerves that hopefully would not go systemic. Typically you can inject something that relatively stays local, that's locally um, you can deliver injections where things stay locally. There are ways you can inject things that go systemically everywhere. And so, doing a subcutaneous injection at these sites where wrinkles typically occur for Botox can help help provide some degree of paralysis that reduces wrinkles and just hoping it doesn't go systemic. Yes.
a chance like money that you can value how some of this works that you get much poisoning or is it
Yeah, great question. So, for certain populations, particularly newborns, infants, and pregnant women, it is suggested, bless you, it is suggested that they stay away from honey because there is those are typically more immunocompromised populations that would not be capable of protecting themselves from exposure to this bacterium. The bacterium carries the toxin and delivers it. Uh, in in immunocompetent individuals, they'd be able to clear out the bacterium fairly quickly. But uh, um, it's in these immunocompromised populations where eating honey would be, particularly raw honey, would be problematic, where they would have exposure to these spores and not have the capacity to protect themselves and clear out the bacterium before the toxin is released and carries out a lot of these symptoms that we talked about. All right, these are great questions. I want to make sure we have time to go through the content and leave room for questions for the content. So we'll go ahead and jump forward. So whenever we talk about enzymes, it's important to consider what an enzyme actually does. So an enzyme is a protein. It is a class of proteins that help catalyze a specific reaction. And when we talk about these reactions, there are certain rates at which these reactions can occur. And so there is a model that was talked about in the pre-lecture that we'll just spend a little time going over in lecture today. It's called the Michaelis-Menten kinetics model, or the Michaelis-Menten model. It helps us understand and predict what the reaction rate is for a given reaction based on particularly the substrate concentration. So when we talk about enzymes and substrates, what we're talking about is the substrate is the molecule, or typically the protein, that an enzyme interacts with and catalyzes the reaction with. And so on both sides of the board, I've kind of written out just like a really brief reaction to help understand how this works. So we have an enzyme plus a substrate. There's a reaction rate at which they produce an enzyme and substrate complex. They're together. And then from there, it'll produce some sort of product with the enzyme still there. And so enzymes will interact with a substrate to produce some product. And the rates at which the reaction occurs, we can model it using this Michaelis-Menten model. With any model, it's a prediction, and there are typically assumptions that undergird this model. And so we'll talk about a couple of those key assumptions, but the model helps us predict what the rate at which this reaction is occurring. And a couple of key parts to this model are first the Vmax. V is the reaction rate, and Vmax is the rate at the saturation point that this reaction reaches to at a given concentration of your substrate. And so typically in this Michaelis-Menten model, just taking a second to point at the board here, if you increase your substrate concentration, again, your enzymes reacting with, if you increase the substrate concentration enough, you'll reach a reaction rate just plateaus, and that plateau is called the Vmax. Okay? And I've drawn on the board kind of at the plateau where that Vmax is. What's important to know is that there's also an important constant that goes with this model called the Michaelis constant. The Michaelis constant, what that is it is the substrate concentration. If you have this drawn out, if the reaction rate, which is usually in moles per unit time, at a Vmax of 10, we'd go down to 5 and look over on the substrate concentration is. That would be our Michaelis constant for this enzymatic reaction. And so there's two important assumptions that go with the Michaelis-Menten model. Excuse me. The first is that it is at steady state, that it reaches a point at which the rate of change over time of the enzyme that the enzyme substrate complex is at steady state, meaning that the rate of change of the concentration of the enzyme substrate is 0. It's constant. Okay. The second is that the concentration of the substrate far exceeds the concentration of the enzyme. Okay. Again, I'm going to repeat these two one more time. The concentration for the enzyme substrate complex is at steady state. Okay. The rate of change of this concentration is zero. There's always a constant, there's no rate of change of this enzyme substrate concentration. The next is that the concentration of the substrate far exceeds that of the enzyme. And that makes sense given that when we are looking at really high substrate concentrations, we expect this to reach a plateau. And so we're working at times with really high substrate concentrations. So to help try and apply this, to take a moment to think through it, if you had to do if you had to do if you had to answer a question about enzyme kinetics and look at and determine a Michaelis constant based on given reaction rates and given substrate concentrations, take a moment to think through what the approximate, again, we're going to be doing lots of back of the napkin calculations in this class. What would you suspect the approximate Michaelis constant to be? Thinking through that the Michaelis constant is the substrate concentration at 1/2 of Vmax. Yes. Sorry? Right, exactly right. It'd be one or just right at one. And so we can look at these reaction rates at this table and see that it reaches its plateau right around 12. So 1/2 of Vmax would be about 6. And so we see at 5.6 that this is at 1. And so if we were to do a back to napkin calculation, we would say like this is just north of 1. And so that is a way to just quickly look at a table and to be able to determine what the Michaelis constant would be. And so when taking a moment to think about actually, you know what, I'll actually take a step back and stop for a couple of questions. Yeah. It, I, it depends. It depends if you see it plateauing out. So if this table, say, went from like 11, uh, 11, 15, 19, it wouldn't quite appear that it was plateauing out yet. So we don't quite know what the Vmax is. So it would be, if this was if I was in a particularly um, interesting mood and wanted to give you a really hard question on a quiz, I would potentially change this to like 11, 15 and say, what would be the Michaelis constant? And the right answer would be, we don't have enough information to determine the Michaelis constant from this table. The key thing to note is that it's not just about what the highest reaction rate is in this table, but it's determining, is it reaching a saturation point? Is it plateauing out? And so if we see a clear point at which it's plateauing, then we can determine what's 1/2 Vmax. And from that, we can determine, based on the substrate concentrations that these reaction rates accompany, we can then determine what the Michaelis constant is. That's a great question. I appreciate you asking that. Yes. Approximately 12 is it would be the Vmax. You can say like, you can potentially look at this and say, since this is the highest, it would be 12.1. But for the sake of a back of the napkin calculation, we would look at this and say Vmax appears to be 12. Does that help make sense?
We just take that plot, the actual.
And this is typically determined experimentally. And so we would see a clear, because this is just one data point, but typically you would see a clear point at could be 12, it could be an integer like that, it could be 13.5, it could be 14.1. But it's rather just the key take-home message here is that Vmax is the point at which the reaction rate plateaus. So again, Vmax is not just just about looking at the table and seeing that it reaches its highest point, but rather where does it plateau off? Because we'll actually talk about later in this lecture how this might change, how these curves might change with the introduction of some sort of molecule. and knowing that the substrate concentration at which we reach 1/2 Vmax is the Michaelis constant. So this is a model that helps us understand um, kinetics and enzyme reactions. All right, so taking a moment to think back to the pre-lecture, if we were looking at two illustrated reactions or reaction rates, uh, which of these do you suspect would happen more easily or happen faster? Yes, exactly right. The one with the curve, the reaction rate with the smaller hump would happen a lot faster because if you think about the energy required to get from reactants to products, this is a lower hurdle for the reaction to occur at B as compared to A. You can try and think of the height of the hump as the energetic obstacle to overcome for this reaction to occur. This is the Gibbs free energy for this reaction to occur. And so it's always helpful. Another like physical illustration that you can think of whenever you see these curves is thinking which curve would it be easier to like push a stone up and over? And so typically it's the hump that has the lowest height will be the easiest reaction to occur. And so what ends up happening is that enzymes typically help a reaction to occur more quickly, going from curve A, introducing the enzyme, it becomes curve B. It becomes more easy for that reaction to occur. So there are classes of drugs that actually target enzymes that are enzyme inhibitors, drugs that treat drugs like Cipro or drugs that help treat HIV infection. There's two major kinds of enzyme inhibitors. We have competitive inhibitors and non-competitive inhibitors. So typically with enzymes, we talked about enzymes having a substrate that come together for the enzyme substrate complex, and for the Michaelis-Menten model, there's no rate of change here. It's at steady state, and it produces a product. So enzymes with their substrate produce a product. Enzymes are destined, for lack of a better phrase, to target a specific substrate and to produce some sort of product. And so in this example here, we have an enzyme that kind of looks like Ms. Pac-Man with a triangular shape site that the substrate binds to. That's called the active site. The site at which the substrate binds to the enzyme is called the active site. There's also a different name for it that we'll talk about in a few slides, but for now, just think of it as the active site. So when a substrate comes and binds to the active site, that catalyzes a reaction, which will produce some sort of product, typically. A key thing to note is that for competitive inhibitors, they bind directly to the active site and compete with the substrate for the active site. So what will end up happening is that we'll have a competitive inhibitor that has a fairly similar structure and conformation that enables it to bind to directly to the active site and slow down the rate of reaction, the catalysis of this reaction. So when we think about a competitive inhibitor, a few key things to note is that it's not changing anything about the enzyme itself. It's competing with the substrate for the active site, but nothing about this enzyme is changing. And so typically Vmax is a feature or quality of the enzyme. So because of that, because we're not changing anything about the enzyme and Vmax is a quality or a characteristic of the enzyme, Vmax does not change. However, the Michaelis constant will. And so because it's competing with that enzyme, what ends up happening, I'm going to have blue be my color for competitive inhibitors. I'll draw a blue square and say competitive inhibitor is CI. So what ends up happening is that at low substrate concentrations, this reaction will happen a lot slower. But what's important to note is that if you increase, so in this direction, we're increasing substrate concentration. If substrate concentration gets high enough, we'll eventually Vmax, because again, Vmax is a quality or feature of the enzyme. We're not changing anything about the enzyme. And so it's just directly competing with substrate. And so again, at low substrate concentrations, the reaction rate is lower. But then eventually, because again, we're not changing anything about this enzyme, we're still going to reach Vmax. And so again, if we push the substrate if we push the substrate concentration high enough, we're still going to reach the same Vmax, the same reaction rate as without the inhibitor, because again, it's competing with the substrate for the active site. But if there's sufficiently high enough substrate concentration, the activity of the competitive inhibitor will be outcompeted by the substrate because its concentration is so high that it'll still reach Vmax. Okay? Now we have our other class of inhibitors, non-competitive inhibitors. It does not bind to the active site. Instead, it binds to what we call the allosteric site. When you hear allosteric or allosteri, think of a loose definition to think of when you hear that phrase, when you hear that word, is to think of something that's working from a distance. Okay? So in this situation, again, non-competitive inhibitors are not binding directly to the active site. Instead, they're binding to a different part of the enzyme. So we have our substrates that still interact with the active site. It's not, it doesn't have to compete with a non-competitive inhibitor for the active site. Instead, what ends up happening is that we have our non-competitive, and it produces a product. Instead, we have our non-competitive inhibitor that binds to the allosteric site, some other region on the enzyme. Now, we talked about this a lot throughout the course of the amino acid and protein lecture. We talked about it a little bit in the cell membrane lecture, but I'm going to try and really harp on this for the next few lectures. Whenever whenever the charge is changed on a protein, it changes its structure, which is going to effectively change part of its function. So here, typically, non-competitive inhibitors will usually change the charge or change the interactions occurring with this enzyme protein. That's going to change its structure, and that's going to change its function. So what ends up happening, I'll take one step back. Once the non-competitive inhibitor is coming in, our active site is triangular. Oh, sorry, wrong direction. There we go. Once it binds, it changes the charge, changes the structure, changes the function. Our active site changes. It goes from, as an example, from triangular to circular now. But our substrate doesn't change. It's still triangular. So because of this, it's changing the actual structure of the enzyme, and it's going to have a lower affinity for the substrate. So because of that, we're changing an actual feature. We're actually changing the enzyme here. Because of that, we're going to change the qualities of this enzyme, and it's going to have a different Vmax. Okay? There's going to be a lower affinity and a lower Vmax because of this non-competitive inhibitor. So I'll have red as non-competitive inhibitor, NCI. And so we're going to reach a different Vmax here. What's important to note is that while we reach the same Vmax, if we push the concentration high enough, we're going to lower Vmax. The important thing to note is that the Michaelis constant largely stays, theoretically, usually, for a non-competitive inhibitor, that enzyme's Michaelis constant, the Km, stays about the same. And so for a competitive inhibitor, we're not changing the enzyme, so the Vmax stays the same. We get to a different Michaelis constant, but Vmax stays the same. For a non-competitive inhibitor, we're changing the actual structure and affinity, and so we reach a different Vmax, but the Michaelis constant typically, in a theoretical sense, stays about the same. Sorry, I just realized I should draw my legend here as well. Another thing to note is that while I talked about the actions of a competitive inhibitor being reversible by increasing the substrate concentration a lot, For a non-competitive inhibitor, there are some that are reversible and there are some that are irreversible. And it usually depends and changes from inhibitor to inhibitor. Yes. It does alter the affinity. Sorry, if I said it doesn't, then I misspoke. Forgive me. Experimentally, yes, but like in a typically a theoretically, let me clarify that. A theoretically perfect non-competitive inhibitor only alters Vmax. Typically, experimentally, we do typically see a different Michaelis constant. But if we had some sort of perfect some sort of perfect non-competitive inhibitor, we would theoretically see the same Michaelis constant, but that's typically kind of to answer your question, that's typically not what we see in biology. Yes. Yeah. Yep.
Five different things like this, like if you have a non-competitive or competitive inhibitor that kind of sticks wherever it keeps the active site or the allosteric site, like almost permanently or for like a long period of time, a millisecond.
Yeah. That's exactly right. So just to try and summarize for the sake of recording, you could have a non-competitive inhibitor and a competitive inhibitor that both target the same enzyme, uh, target different parts of the enzyme, the competitive inhibitor on the active site, the non-competitive inhibitor on the allosteric site. And it could be either a permanent inhibition in the case of a non-competitive inhibitor. It could be a temporary inhibition for a competitive inhibitor, as long as depending on the concentration of the substrates and depending on what kind of non-competitive inhibitor it is. Yeah. Sorry? Yeah, so that goes back to the phrase. A phrase. I'll put like a like digital asterisk here. A phrase I'm going to keep coming back to is if there's a modification that changes the charge, it's going to change the structure and it's going to change the function. So for a non-competitive inhibitor, they'll usually have some sort of charge with them that changes the structure of the enzyme, and that's going to change the function of the enzyme. It changes the active site, that's going to make it have a lower affinity for the substrate. Okay, I'm going to make that, I'm going to say this again for the sake of the recording. Non-competitive inhibitors actually change the structure of the enzyme so that the active site has a lower affinity for the substrate. Did you have a follow-up? This is taking a moment to think about only a non-competitive inhibitor present here. If there was a non-competitive inhibitor and a competitive inhibitor together simultaneously, the competitive inhibitor would have a reduced efficacy if there was also a non-competitive inhibitor there as well. But it's not. But there would still be some degree of inhibition provided by the competitive inhibitor. But for the sake of making sure that the take-home message is clear, non-competitive inhibitors, because it sounds like I didn't do a good job of articulating the change in affinity, non-competitive inhibitors change the structure of the active site and lower the affinity for the substrate. Got a few questions here. Yes.
Nicholas constant, the point at which it's half of the constant velocity.
Yes, but it's sorry, the Michaelis constant is not half the maximal velocity. It's the substrate concentration at which we reach half maximal velocity. So because it's the substrate concentration, it theoretically could still be the same if even though the Vmax changes. Because, for example, say this is at 20, we go down to 10 and see the substrate concentration at which it occurs. If this is at 8, we drop down to 4. At 4, theoretically, we could still be at the same, we could theoretically still have the same Michaelis constant because it's the substrate concentration which we reach when have Vmax. I'm going to go here, then here. Go ahead. So it's a key constant for a given enzyme that helps us model the reaction rate. It ties into the Michaelis-Menten model, and so it's just a it's a key figure that we calculate that enables us to use that model. Yes. Yeah. Yeah.
Yeah, so if there was already a substrate binding to the enzyme active site and then not competitive with its constant site on the same enzyme, would it still, I guess, change the structure and function of the enzyme?
If just to make sure I'm tracking, if a substrate is already bound to the enzyme, so like say we have our enzyme substrate complex, non-competitive inhibitor comes in, is your question then basically like what happens?
Yeah, does it still change?
Yeah, it will. So in that situation, there's still going to be a change that then lowers the affinity for those interactions to occur between enzyme and substrate. And so in that situation, they'll functionally disassociate and you'll have less product formed as a result. Yeah, great question. Go ahead and then we're going to keep moving forward. Yeah, so there's a lower, so it's going to bind to it at a lower rate or lower degree. So we have the same, again, like we have some of the same amino acids in that general area. However, the conformation has changed. When we think about active sites, we sometimes physically will refer to them as like a binding pocket. So you can almost think of it as like a key and lock mechanism, where you can almost think of I don't know if I'm aging myself here, but thinking back to like using an old key to get into a door, um, the grooves have probably shifted and changed over time. And you're going to be less successful opening the door as opposed to like a clean brand new key that does it right every time. So you can think of this as a really old key now that has a hard time unlocking and opening the door because there's not quite the same structure that's meant to nicely fit the binding pocket of this enzyme. And so similarly, you're still going to have substrate able to bind loosely to the active site and still produce a product, but it's going to have a lower affinity. It's going to happen less frequently. Does that help? All right, let's keep marching forward. I think we'll be able to finish everything today. And I promise not to just like fly through everything like I did at the end of the last lecture. So we talked about allosteric, meaning working from a distance. So there is a key regulation that is provided at the allosteric site for enzymes. So when we talk about allosteric regulation, that's when some sort of molecule, we'll usually refer to it as a regulatory molecule, that binds to a site separate from the active site. That is allosteric regulation. Again, it will provide a charge that changes the structure, that changes the function. This allosteric regulatory site will alter the shape of the enzyme and change its activity, change its affinity for the substrate. Now, so far, we have only talked about, we'll call it negative regulation, negative allosteric regulation, where, for example, again, the non-competitive inhibitor binds to the allosteric site and changes the active site and lowers the affinity. You can also have positive allosteric regulation, where some sort of regulatory molecule binds to an allosteric site, changes the structure of the active site, and increases the affinity for the substrate. So now you're going to have better binding and better interactions between the substrate and the enzyme. So far we've referred to our sites as the active site and the allosteric site. Other names that you can hear for these enzymes are the catalytic site, that's the active site, and a good way to think of it is excuse me, my voice cracked a little bit. A good way to think of it is it's a site or domain that catalyzes the reaction. Another name for the allosteric site is the regulatory site. And so you'll typically also not hear it as site, but as domains. We have the catalytic domain, that's the active site, and we have the regulatory domain, that's the allosteric site. So again, we can have positive and negative allosteric regulation. Positive would increase the affinity of the active site for the substrate, and negative allosteric regulation would lower the affinity of the active site for a substrate. And again, catalytic domain is another name for the active site. Regulatory domain is another name for the allosteric site. But I'm going to take a moment to try and describe when we say the word domain, this is really important. So we've talked about subunits already. Now we're talking about domains. I want to try and take a moment to try and make sure that we reconcile these two phrases, these two words, so that we understand them in the right context when talking about proteins. When we talk about domains, we are talking about different parts of one protein. Okay, we're talking about a single polypeptide where there's different parts of that one protein that we refer to as domains. So some given single protein can have a region that's called domain A and domain B. And so to continue to use dry erase markers as my example, we could call this the cap domain and the backend domain. Sorry, I was trying to think of a better phrase, but that's what we'll just say for now. But this is one single protein, okay? But there's different regions of it that we'd refer to as domains. I'll stick with cap domain. I like that. I'm gonna stick with just that for now. It's funny when you catch things on recording that you don't mean to. On the other side, we also have subunits. So we continuing the the dry erase marker example. Subunits are, and I'll take one step back. And so domains are also illustrated with a single given tertiary structure. Subunits are two separate proteins that come together to form a protein assembly, a multi-protein assembly, and each single protein of that protein multi-protein assembly are called subunits. So we'd call this the black subunit, the red subunit, and the blue subunit. Each of them is a single protein on their own, and each of them have their own domains. Domains are part of a single protein, but it is possible that these proteins can come together, form a larger protein structure, a protein assembly, where each one of these individual proteins is referred to as a subunit. So a subunit is its own protein. It is a full protein that has its own gene. Each subunit has their own gene that are associated with them. Not all proteins will do this, but in some cases, proteins can come together to form a larger protein assembly, where each single protein, again, has their own domain region sometimes. But when they come together, each of them are referred to as a subunit. So domains are parts of a single protein. Subunits are separate proteins that are part of a larger structure together. So to understand why we're talking about this, when we get to the botulinum toxin, there are different domains. So because it's domains, it's one protein, one single polypeptide. And so we have the receptor binding domain, we have the translocation domain, and catalytic domain. These different domains are typically identified based on some specific function that they provide. You see above that there's light chain and heavy chain labeled. That's typically identified when we're experimentally characterizing these proteins and running them down a gel. And when you run them down a gel, you'll see two chains sometimes emerge at certain molecular weights. And so that's typically, if you see light chain, heavy chain of some protein, it's a experimentally determined thing that you see when you run them on a gel. And we'll talk about running proteins on a gel during our protein biochemistry lecture. So if that doesn't make sense now, it will, hopefully, when we get to that lecture. But the key thing to note here is that domains are part of a single protein, and each of them imbue the protein with a certain function. When we talk about taking a second to pivot back to regulation, allosteric regulation, there's different modifications that can happen that allosterically regulate an enzyme. We can have covalent modification by the addition or removal of certain groups. One example of that is the addition or removal of a phosphate group. There is a class of enzymes whose entire function is to add phosphate groups. Those are called kinases. And so, for example, if a kinase comes in to add a phosphate group to this enzyme, the addition of that phosphate group, it carries a charge. They're negatively charged. That changes the structure. That changes the function. So now this enzyme, by the addition of this phosphate group, is now typically now it's quote unquote activated and ready to produce product when it interacts with its targeted substrate. There's also a class of enzymes that remove phosphate groups. Those are called phosphatases. So we're going to start seeing the names of some enzymes. The key thing to note is that there's always a suffix that denotes that a protein is an enzyme. It's going to be ASE. And typically what comes before that suffix of ACE, again, ACE means that it's an enzyme, typically denotes what it targets. There are proteases, just large swath of class of enzymes that just target proteins in general. There are lipases, enzymes that target lipids. We just talked about kinases. One thing to note is that there are different amino acids that are specifically that are the sites at which a phosphate group is added. The addition of a phosphate group, we call it phosphorylation. So the amino acids where phosphorylation occurs are on serines, threonines, and tyrosines. There is a class of kinases that specifically phosphorylated tyrosine that's called tyrosine kinases. Hopefully these names are going to start getting a little simpler and you guys can appreciate them. Yes. Great question. So there are sometimes going to be different allosteric sites depending on the molecule that's providing that regulation. So for example, this addition of a phosphorylation group is allosteric regulation. It's not binding to the active site. It's binding to some phosphorylation site that's a serine, threonine, or tyrosine. Typically, there are many sites at which phosphorylation can occur on some given enzyme. And so what's going to end up happening is that there might be different kinds of regulation that are provided. This is actually a great question because this is like an area of active research. So depending on the kind of phosphorylation, so we talked about for the sickle cell lecture that they're at the USA at the sixth amino acid, there's a valine substitution. So for example, typically for phosphorylation, I'm just going to nerd out for a hot second, so please forgive me. So at some specific amino acid, say amino acid number 73 is a serine, it can be phosphorylated there. That phosphorylation event imbues some sort of change to the enzyme that could increase its or decrease it. Let's move down to amino acid number 131. That's a so that it can be phosphorylated, and that at that different part, there could be a different change that happens to the enzyme that could increase its activity or decrease it. So there are different allosteric sites for a given enzyme, depending on this kind of regulation. For phosphorylation, it happens all over. It can happen all over. When I say all over, there's probably, depending on the size of the protein, maybe 4 or 5 known phosphorylation sites. And so these different phosphorylation sites could alter it somehow differently based on these different phosphorylation events. But I'm going to step back and reorient myself to make sure that there's a clear take-home message here. There are potentially multiple allosteric sites on a given enzyme. And those different allosteric sites could each lead to a different kind of regulation of the enzyme. Yes, we would call that phosphorylate. We would say at this specific phosphorylation site where that phosphate group is, that is allosteric regulation because in this hypothetical situation, it is increasing the affinity for the substrate. So we will say that phosphorylation event at that site is an example of positive allosteric regulation. There could be a different phosphorylation site that changes and decreases the affinity and is an example of negative regulation. But at that site, that's an example of positive allosteric regulation. Does that help clarify? All right. Sorry, let me get my bearings for one moment. Okay. So we have the addition and the removal of groups. We can also have the cleaving of bonds. typically when a protein is synthesized, it's formed in a precursor state with this little cap on the end. We usually call it the pro-cap. Um, and so for enzymes that are in their precursor inactive form, they're at times in their pro-enzyme form. And so there can be the cleaving of bond, that a cleaving of that cap that removes it. And again, I'm going to keep going to the dry erase marker. It's not functional yet. We have the pro-cap. This works perfectly, you guys. I know this is sometimes a little dry, but I'm really appreciating that this is actually working well. If you remove the pro-cap, it's now active. I can now use it. If the pro cap is still there, it's not going to be quite active. I appreciate your smiling because I needed someone to smile to give me some positive reinforcement. I see you. I see you. An example of this is thrombin. So the clotting cascade is a great example where cleaving of bonds activates enzymes that help prepare them to target their substrate and produce a product. We also have the association and dissociation of different subunits that can lead to some sort of regulation of an enzyme. So again, this is subunits. So this is two different full proteins that can either come together or separate that provide regulation. So we talked about how there are kinases that add phosphate groups to proteins. There are tyrosine kinases where they specifically add it to a tyrosine residue. When we say residues or motifs, we're talking about specific amino acids. There is a class of tyrosine kinases called receptor tyrosine kinases. So typically, and we talk about receptors are these proteins that are on the cell membrane. When they, again, this is like a lock and key mechanism. When they bind to their ligand, a ligand is something that binds to a receptor, there can be the association of two subunits of a receptor that come together, and underneath there's a receptor tyrosine kinase that targets some substrate, leads to a product, and leads to downstream signaling inside the cell. And so the key thing to note on this bullet point is that there can be the association and dissociation, the movement of some of these different subunits that can lead to the activation of some enzyme. So with receptor tyrosine kinases, these subunits come together when they bind to some ligand. Again, I'm going to keep using the marker. If the marker is the ligand and my hand is the receptor, these two subunits come together because they're both binding to the ligand, this marker, and by the association bringing in, there's a part here that's an enzyme, a receptor tyrosine kinase, that now is ready and activated to produce a product from the substrate. Finally, we have something that's called cooperativity. This is something that's really unique where the product that's formed from this reaction, okay? So we have an enzyme, substrate that produces a complex that produces a product here. Cooperativity is when the product promotes the formation of more product. It feeds back into this reaction and supports and speeds up this reaction a little bit more by enabling it to produce more product. And so this can imply that some given enzyme might have multiple catalytic subunits, multiple regions at multiple active sites where it can interact with some given substrate. And so given that there are different degrees of cooperativity that can happen, different degrees at which a product can enhance the formation of more product, it is actually incorporated into the Michaelis-Menten equation by the coefficient n as an exponent here, and so we call that coefficient there the Hill coefficient. The Hill coefficient in the Michaelis-Menten equation here, helps to indicate how much cooperativity is occurring for this specific reaction where cooperativity is at play. And so looking at where we have an n of 1 for the cooperativity, which would be kind of like your standard Michaelis-Menten model, as we increase the degree of cooperativity, we have a higher Vmax, higher speed, which a higher reaction rate that we can reach for some given reaction via the Michaelis-Menten model. An example of this cooperativity, we have an enzyme that's called glycogen synthase kinase 3. So really big name of an enzyme. We can break it down. It's a kinase, so what does that mean it does? Adds a phosphate group. It's a glycogen synthase kinase. What do you think it's phosphorylating? Glycogen, it phosphorylates glycogen synthase. Taking a moment to look at glycogen synthase, that it has, so it's phosphorylating glycogen synthase. Glycogen synthase itself, it has ASE on the end, it has ACE. That means it's an enzyme. That enzyme's function is to synthesize glycogen. Okay? And so the reason that there is cooperativity here is because the motif that glycogen synthase kinase 3 targets is when there is a serine. When we see X, looking at these at the amino acid sequence, that's just any old amino acid. Could be anything. So when we have a serine, 3 amino acids, and then a phosphorylate, that is the motif that GSK3 enzyme up there targets, and it phosphorylates the serine. What ends up happening is that we have what we call a priming phosphorylation event by casein kinase 2. That's CK2. So we have our first priming phosphorylation event here. And then G3 can see serine, 3 amino acids, and then a phosphorylated serine. Let's phosphorylate this. Now the product has become the substrate again because with this phosphorylated serine, we have 3 amino acids and another serine. And then phosphorylation event again. So it turns out we have another 3 amino acids and another serine. So this is an example where the product itself is now becoming the substrate. Okay? It's able to continually phosphorylate based on the amino acid sequence and what it targets, that it can continue to catalyze this reaction of phosphate groups continuing to be added to the phosphorylation that's happening on glycogen synthase here. And so GSK3, it's a kinase that adds phosphate groups, specifically adds phosphate group serine. There's 3 amino acids and a phosphorylated serine there. It can continue to phosphorylate. That's the reaction being catalyzed by that enzyme, GSK3. And this, once the first phosphorylation event happens, this becomes the product. This product, again, because of the sequence, it becomes a substrate again, and it continues over and over again because the substrate is the product is becoming the substrate. This is an example of cooperativity here. Are there any questions about cooperativity or anything we covered here? Yes. Motif just means the amino acid sequence that is the target. So when we hear residues or motif, think amino acid level. Okay? When you hear residues or motifs, think amino acid level. So the motif or the amino acids that is the substrate for GSK3, again, this is just an example of cooperativity. What it targets is a serine, any 3 amino acids, and a phosphorylated serine. When you see a lowercase p in front of a serine, threonine, or tyrosine, that means it's phosphorylated. Okay? So this serine is where the phosphate group is going to be added by GSK3. Once that happens, if we take a look at the amino acid sequence, we can see product after we add this phosphate group here, or not we, GSK3 does. All of a sudden, that product has become the substrate again because now that this phosphorylated serine kind of becomes this one over here based on the amino acid sequence. We can look over and see here's another serine, another 3 amino acids, phosphorylation event happens again. Look over, phosphorylation event happens again. Look over, phosphorylation event happens again. This is where the product is becoming the substrate. Okay? Cooperativity is largely defined as the product helping with the formation of more product. This is a specific example where the product itself is becoming the substrate. Yeah. Yes, typically with cooperativity, because it's helping with the formation of more product, that's how we define cooperativity, it's going to increase the rate of the reaction. Okay? I saw like a line of back there, right? Or sorry, yes, you. Go ahead. I'm going to learn names eventually, but forgive me if I say you go. The product is a phosphorylated serine. Going back to a question I had earlier, that this this phosphorylation event can have an impact on this enzyme glycogen synthase to potentially produce more glycogen. So these phosphorylation events, in this case, just looking at the enzyme, the product is phosphorylated serine. This phosphorylation event, that's the product. Um, that is the reaction that's being catalyzed by GSK3. And what it's potentially doing, what it's likely doing, is increasing the reaction that's being catalyzed by glycogen synthase itself to, again, looking at the name, synthesizing glycogen to potentially produce more glycogen, to catalyze a reaction that produces more glycogen. Does that help clarify? Go ahead. If the reaction just keeps happening over again, it would reach a Vmax if the substrate concentration was incredibly high. There is, trying to not go too in the weeds here, these phosphorylation events, these addition of phosphate groups, there's turnover. So we talked about kinases that add phosphate groups. There are phosphatases that remove these phosphate groups. So continuing to use this as an example, there's turnover where eventually over time, and we talk more about glycogen in our very last unit on metabolism, but the ability to produce glycogen is important. So there's going to be turnover of this enzyme where it's turned off if there's not a need to produce more glycogen, or turned on if there's a need to replenish our glycogen stores. Just kind of using this as an example, but there's just turnover of it. If that helps clarify. We have 5 minutes left, and I think I can get us there. So I don't appreciate the laughter. All right, so because I'm not a neuroscientist, I'm going to take a moment to play this video that helps set the foundation of what we're going to finish with. And here we go.
Neurons begins when an electrical impulse called an action potential travels along the axon of a presynaptic neuron toward the axon terminal. The action potential cannot cross the synaptic space. When it reaches the axon terminal, it causes membranous sacs called vesicles to move toward the membrane of the axon terminal. The membrane of the vesicle fuses with the membrane of the axon terminal, enabling the vesicle to release its contents into the synaptic space. The molecules released from the vesicles are chemicals called neurotransmitters. They drift across the synaptic space and bind to special proteins called receptors on the postsynaptic neuron. The binding of a neurotransmitter to its receptor can trigger an action potential in the postsynaptic neuron. That electrical signal then moves toward the cell body of the postsynaptic neuron. Now that the neurotransmitter has relayed its message, it releases from the receptor into the synaptic space. Some of the neurotransmitter is degraded by enzymes in the synaptic space, and some of the neurotransmitter is carried back into the presynaptic neuron through transporter proteins. The neurotransmitter that is taken back up into the presynaptic neuron is then repackaged into vesicles that can be released the next time an action potential reaches the axon terminal. The entire process repeats each time an action potential reaches the axon terminal of the presynaptic neuron.
So the key thing for us to note here is that these vesicles that have their own membrane, that is comprised of a phospholipid bilayer, when they meet in our cell's membrane with its own phospholipid bilayer, the hydrophobic interactions, the energy required to overcome the hydrophobic interactions that keep the phospholipid bilayer in place is an incredibly high amount of energy. So to be able to fuse together these two membranes to release the contents of a vesicle requires a lot of energy, but is also incredibly important for our cells, some of their key fundamental functions. For the sake of a neuron, releasing neurotransmitter, for the fundamental function of a lot of cells, the exocytosis, the exit of contents of a vesicle, or sometimes endocytosis, the taking in of contents that are outside into the cell. And so the way in which that happens, or the way a vesicle actually really looks like, is there's a phospholipid bilayer with all of these proteins littered all over the surface. The reason why we're talking about this is because we're going to come back to a specific protein on the vesicle surface called synaptobrevin that is really important for botulism poisoning. So to set the stage here, the way that these high energies are overcome for the vesicle membrane and the cell membrane to fuse together is that there's a class of proteins called snares. Okay, they enable they enable the fusion of these two membranes. We have two kinds of snares. We have, excuse me, vesicular snares or V snares. Those are the snares that are on the vesicle membrane, and there are T snares. In this case, those are the snares that are on the cell membrane. What ends up happening is that the V snare on the vesicle and the T snare in the cell membrane, once the vesicle membrane comes up into contact, once the vesicle comes into contact with the cell membrane on the inside, the V snare and the T snare actually have a coil-coil interaction where they start to wrap themselves around each other and actually start to push out every water molecule out of the way to enable for the fusion of the two phospholipid bilayers. The coil-coil interaction is pretty remarkable. If you think about a large like tug of war rope, and if you start to coil it, think about like how it all of a sudden just like loops and kind of like snaps into a coil itself. That's kind of what happens with the V snare and T snare as they coil around and fuse together the vesicle membrane with the cell membrane. And so what ends up happening is that synaptobrevin is a V snare found on the synaptic vesicles of that contain neurotransmitters. So the really important vesicles that contain neurotransmitters, that the V snares that we're going to focus on is synaptobrevin, and the botulinum toxin that comes from that Clostridium botulinum bacterium. It is an enzyme that targets synaptobrevin. It cuts it and inactivates it. So what ends up happening is the bacterium, whether you're eating raw honey as an immunocompromised individual, eating poorly cleaned food, eating dirty soil, the bacterium will enter your gut lumen. And this is also another area of active research where just 2 years ago, the mechanism of how this happened was characterized before the toxin was described to somehow cross your gut lining. That's called your intestinal epithelium. Anywhere there's like a lining, there's usually an epithelial cell sheet. That's called an epithelium. So your gut epithelium, it was somehow determined this toxin crosses it. We've now identified the specific kind of epithelial cells and why it happens there. The toxin crosses your gut epithelium, enters the bloodstream, and somehow eventually gets to a motor neuron where it can then target synaptobrevin, cut it, and render that synaptic vesicle non-functioning. Over time, that toxin will continue to catalyze the reaction and cut synaptobrevin on every synaptic vesicle within a given motor neuron. Now, if it's in the bloodstream, how does it get to a motor neuron? When we get back, I just said all this, so I'm just going to fast forward. And how does it get to a motor neuron where it's internalized by the motor neuron by endocytosis? There's a receptor binding domain of the botulinum toxin that targets motor neurons specifically. So once it's endocytosed, taken in by a motor neuron, and it's in a vesicle that's been endocytosed, the next thing that happens is quite remarkable. This translocation domain starts to take over. What I mean by that is the pH, usually when something's endocytosed, if it's not something that is known to the cell, it'll prepare the contents to be degraded by decreasing the pH, making it way more acidic. Now, we've talked about this. If you change the pH, you're potentially going to change the structure of that protein and change its function. In this endosome, where the pH is being lowered, this protein changes its structure, and the translocation domain becomes a harpoon where it shoots out of the endosome and gets into gets inside the cell, but is outside of that endosome where it's going to be destroyed. Once that happens, this catalytic domain can now function to target synaptobrevin and destroy every synaptobrevin molecule that it can find and interact with. Again, yeah, the toxin escapes. The light chain or the catalytic domain is a zinc endopeptidase. So peptidase, it's an enzyme that targets a peptide, a protein. Endo, it cuts it in the middle. So it cuts between the catalytic domain and the translocation domain, and it requires zinc. So you can look at any enzyme and slowly break down what its function is based on the name. And so what ends up happening next is that if there's no synaptobrevin, you're not going to have vesicles fusing to release neurotransmitters. You're not going to have neurotransmitter release. That's going to lead to paralysis. What's really noteworthy is that the toxin can keep catalyzing the reaction over the course of days. When we ask ourselves how much damage can it actually do, what's important to note is that one single molecule of the botulinum toxin, in a few hours, it can kill a single neuron. And so over the course of multiple days, there's an ability for paralysis to occur. There is a practice question to define what the turnover rate is. That's something we talk about in the pre-lecture. That's something we can talk more about at office hours, but that's just defining the number of reactions per second for a given catalytic domain. And we'd be able to define that a single toxin can destroy a neuron in a matter of a few hours. The treatment are antitoxins. Those are antibodies against the toxin that neutralize it. There's nothing to be done for the motor neurons that have already been destroyed, though. Antibiotics is another option to clear out the bacteria that leads to the release of the toxin, and respiratory support for patients. If bacterial toxins or membrane fusion really fascinated you, here's a few folks at UVA in this space. Sorry for going 5 minutes over, and thanks for your time.